Hello everyone and welcome to the new session of the lecture series, and today we'll be discussing the NEET previous year questions from the topic photosynthesis in higher plants. Okay, so let's start with the first question, which is how many ATP and NADPH2 are required for the synthesis of one molecule of glucose during the Calvin cycle. Okay, so glucose is a 6-carbon compound. It is C6H12O6. Okay. Now, we know that for fixation of one carbon dioxide molecule, 3 ATP are required and 2 NADPH2 are required. Okay. So, out of the 3 ATP, 2 ATP are used where? During the reduction phase. Okay. And 1 ATP is used where? During the regeneration of the RUBP. Okay, and 2 NADPH again are used in the reduction phase. Okay, in reduction phase what happens? 3-phosphoglyceric acid is converted into 3-phosphoglyceraldehyde. Okay, so for one carbon dioxide, 3 ATP and 2 NADP is used. For fixation of 6 carbon dioxide to form one molecule of glucose. Okay, we can multiply it with 6. So 18 ATP and 12 NADPH2 will be required. Okay, so these are the options. What should be the answer here then? Answer should be 18 ATP and 12 NADPH2. Alright, so let's start with the next question, which is we have to match list 1 with list 2. List 1 consists of the photosynthetic pigment and list 2 is the color of the pigments. Now chlorophyll A is, we know, is blue-green in color. So A is 3. Okay, chlorophyll B is yellow-green in color. Okay, xanthophylls are yellow in color. And carotenoids are yellow to yellow-green in color. Okay, so A is 3, B should be 2nd, C is 4th, and D is 1. Okay, so this should be the answer. So these are the options here. Okay, what should be the answer? Option A is the correct answer here. The next question is phosphoenol pyruvate is the primary carbon dioxide acceptor in. Okay, so if we talk about the C4 plants. So in C4 plant, what happens? Carbon dioxide is accepted by the primary acceptor known as phosphoenol pyruvate. Okay, which is a 3-carbon compound. Carbon dioxide contains 1 carbon and this leads to the formation of a 4-carbon compound, oxaloacetate. Okay, so phosphoenol pyruvate is the primary carbon dioxide acceptor in. Answer should be C4 plants. Okay, this was a very straightforward question. The next question is the water vapor comes out from the plant leaf through the stomatal opening. So here they are talking about the process of transpiration. Okay, so there is a water loss from the stomata they are saying. Now they are saying through the same stomatal opening, carbon dioxide fixation or carbon dioxide diffuses into the plant during the photosynthesis. Okay, now they are asking that reason out the above statement using one of the following options. Okay, so these are the options. First one, the above process happen only during the night time. Okay, which is not true because stomata are also open during the daytime and gaseous exchange also takes place during the daytime. Okay, daytime also there is a loss of water also. Okay, the next option is one process occurs during daytime and other at night. So this is also not true. This can happen anytime. Now third option is both processes cannot happen simultaneously. Not true. It can happen simultaneously. Now the fourth option is both processes can happen together because the diffusion coefficient of water and carbon dioxide is different. Okay, so option D is the correct answer here. Both processes can happen together. Okay, why? Because of the difference in the diffusion coefficient of water and carbon dioxide. The next question is, a plant in your garden avoids photorespiratory losses, okay, and has improved water use efficiency, and it also shows high rates of photosynthesis at higher temperature. This you have to remember, at higher temperature they are saying. Okay, and it also has improved efficiency of nitrogen utilization. This also you can underline. Okay, now in which of the following physiological groups would you assign this plant? Okay, now options are CAM plants, nitrogen fixer, C3 plants and C4 plants. Okay, so if we talk about the C4 plant, it avoids respiration. Okay, first point is avoid photorespiration. So this is true about the C4 plants. They have the Cran's anatomy. That's why they avoid the photorespiration. Has improved water use efficiency. Okay. They also have improved water use efficiency. This is also true about the C4 plants. Okay. Show higher rate of photosynthesis at higher temperature. Okay. This is also true for the C4 plant. Okay. Higher rate of photosynthesis. Okay. And then they are efficient. Okay. They are also efficient in nitrogen fixing. Okay. Nitrogen fixers. There are good nitrogen fixers also. Okay. So what should be the answer here? Answer is option D, C4 plants. The next question is, the process making major difference between C3 and C4 plant is, first is respiration. No, this is common to both. Okay. Then we have Calvin cycle. Calvin cycle also is common. Calvin cycle occurs in both C3 and C4 plants. Then we have photorespiration. Okay, photorespiration. C3 plants, okay, have or they show or they carry out photorespiration. Alright, whereas C4 plants, they have reduced or less or no photorespiration. Why? Because of the presence of the unique leaf anatomy known as the Cran's anatomy. Okay, due to what? Due to the presence of Cran's anatomy. Okay, and last option is glycolysis. Glycolysis is also common to both. Glycolysis means what? Breakdown of the glucose molecule. Breakdown of glucose molecule, and it is a pathway in respiration. Okay, so glycolysis is breakdown of glucose and then it releases energy also. So what should be the answer here? Answer should be photorespiration. The difference between C3 and C4 plants is the photorespiratory pathway. The next question is, when one carbon dioxide molecule is fixed as one molecule of triose phosphate, triose means a compound containing 3 carbon. Now, which of the following photochemically made high energy chemical intermediates are used in the reduction phase of what? Of the C3 cycle they are talking about. So C3 cycle is divided into 3 phases. First is the carboxylation phase. Okay. Then we have the reduction phase. And then we have the regeneration phase. Okay, so they are talking about the reduction phase here. So what happens during the reduction phase? 2 ATP are used and 2 NADPH are also used. So let's talk about reduction phase now. So in carboxylation phase, we know that carbon dioxide combines with RuBP and results in the formation of 3-phosphoglyceric acid. Okay. Now what happens in the reduction phase? There are two chemical reactions that take place in the reduction phase. First reaction is when 3-phosphoglyceric acid is converted into 1,3-bisphosphoglyceric acid. Here ATP is used. Okay, ATP is converted into ADP. Alright, now 2 molecules of 3-phosphoglyceric acid are produced here. Okay, so 2 ATP will be used and 2 molecules of 1,3-bisphosphoglyceric acid will be produced. Now another reaction that takes place during the reduction phase is this 1,3-bisphosphoglyceric acid which is produced is now converted into 3-phosphoglyceraldehyde. Okay, aldehyde group. We have aldehyde group now from the acid group. So this is known as reduction. Here what happens? Again 2 molecules were present here. So 2 molecules will be synthesized here. And what happens here? 2 molecules of NADPH2 will be used and 2 molecules of NADP will be produced. Okay. So you can see here for the fixation of 1 carbon dioxide molecule in the reduction phase, if we talk about, now 2 ATP are used. Okay, here 2 ATP are used and 2 NADPH plus 2 NADPH are used. Okay, so we have the options here 2 ATP and 2 NADPH, 1 ATP and 1 NADPH, 1 ATP and 2 NADPH, 2 ATP and 1 NADPH. So answer should be 2 ATP and 2 NADPH. The next question is, in Cran's anatomy, the bundle sheath cells have, okay, they have thick walls, many intercellular spaces. Not true. Bundle sheath cells don't have any intercellular spaces. Next is thin walled and no intercellular spaces. Okay, thin walled. This is not correct. They have thick walls. Now, third option is thin walled. Again, not correct. And many intercellular spaces, not correct. And no chloroplast is also not correct. Fourth option is thick walls, no intercellular spaces, and large number of chloroplasts. So these are the feature of the bundle sheath cells. Okay, so what should be the correct answer? Option D is the correct answer. Now coming to the next question, which is phosphoglyceric acid as the first carbon dioxide fixation product was discovered in the photosynthesis of, okay, first is algae, second is angiosperm, third is gymnosperm, and then we have bryophyte. Okay, so initial experiments were done in what? In algae. Okay, initial experiment done in algae, and it was discovered that phosphoglyceric acid is the first carbon dioxide fixation product. Okay, so here they are talking about PGA, means they're talking about the C3 cycle. In C3 cycle, what happens? Carbon dioxide combines with RuBP, okay, and it leads to the formation of 3-phosphoglyceric acid. Okay, and which enzyme is involved here? Rubisco. Okay, so algae is the answer. Option A is the correct answer. The next question is, Cran's anatomy is one of the characteristic of the leaves of, okay, now we have the options potato, wheat, sugarcane and mustard. Okay, now if we talk about, let's talk about the crane's anatomy. Okay, so crane's anatomy, first of all, it is a unique leaf adaptation. Okay, seen in what? Seen in plants that are, okay, or that reside in what? Plants that live or reside in tropical conditions. Okay, means dry conditions. So what happens in crane's anatomy? Two types of photosynthetic cells are present. First is mesophyll cells and the next other one is the bundle sheath cells. So why it is known as cranes? So cranes means a ring. Okay, meaning of cranes is ring or a wreath. Okay, so what happens? The mesophyll cells and the bundle sheath cells, okay, which are the two types of photosynthetic cells. And bundle sheath cells are clustered around the vascular bundles. They are clustered around the vascular bundles. Alright, they form a wreath or a ring. Okay, so options we have already discussed. So what should be the answer here? We are talking about the C4 plant here. Okay, and which is a C4 plant? Sugarcane is a C4 plant. The next question is, electrons from excited chlorophyll molecule of photosystem 2 are accepted by, okay, options are cytochrome F, cytochrome B, pheophytin and ferredoxin. Okay, so now we know if we talk about the non-cyclic photophosphorylation. Okay, non-cyclic photophosphorylation. So what happens? Both photosystem 1 and photosystem 2 are involved. Now photosystem 2 absorbs the sunlight, it gets excited, and here what we have, primary receptor is pheophytin. Primary electron receptor is pheophytin here. Okay, and then the electrons move down hill to photosystem 1, and photosystem 1 is also excited, okay, by absorption of the photons. Here the primary receptor is FRS, ferredoxin reducing substance. We transfer the electrons to ferredoxin. Okay, so what should be the answer here? Pheophytin. Pheophytin is the electron receptor. The next question is, in photorespiration, glycolate is converted into serine and carbon dioxide in, okay, so what they are talking about, the photorespiratory pathway now. Alright, so photorespiratory pathway involves the coordination between three organelles: mitochondria, chloroplast and peroxisomes. Okay, vacuoles are not involved. So here they are talking glycolate, okay, which is a 2-carbon compound. That's why photorespiration is also known as C2 cycle. Okay, so glycolate is converted into serine and carbon dioxide in which organelle? Okay, so this is a very straightforward question. The answer is mitochondria. Alright, in mitochondria, this decarboxylation step takes place. Okay, decarboxylation results in the formation of carbon dioxide. Okay. Now, next question is, given below are two statements. First statement is Rubisco is the most abundant enzyme in the world. Okay, so this statement is true. Okay, it is correct. Next statement is photorespiration does not occur in C4 plants. So this is also true. Okay, C4 plants avoid photorespiration. Why? Because they have the Cran's anatomy. Alright. Due to Cran's anatomy, what happens? There is a spatial separation of the carbon dioxide fixation. Or you can say spatial separation of carbon fixation. Okay. And what else? There is a concentration of carbon dioxide in the bundle sheath cells. Okay, carbon dioxide is concentrated in the bundle sheath cells. Okay, so what happens? Due to the concentration of this carbon dioxide in bundle sheath cell, Rubisco will bind to carbon dioxide instead of oxygen. Alright, and thus avoid photorespiratory pathway. So both these statements are true. So in the light of the above statement, we have to choose the most appropriate answer from the options given below. First option is statement 1 is correct, but statement 2 is incorrect. So this is not the answer. Next is statement 1 is incorrect, but statement 2 is correct. So this is not the answer. Both statement 1 and 2 are correct. Okay, so both statements were true. So this should be the answer. And the fourth option is both statement 1 and 2 are incorrect. So this is also not true. So what should be the answer? Option C is the correct answer here. The next question is the ratio of carbon dioxide fixation between C4 and C3 plants are. Option are 2:1, 2:3, 1:1 and 1:2. Okay. So this is again a straightforward question. The answer to this is option A, 2:1. So what happens? C4 plants are, if you talk about C4 plants, they are more photosynthetically efficient. Okay? More photosynthetically efficient. Why? Because they avoid photorespiration. They avoid the process or the pathway known as photorespiration. Alright. So there is no loss of carbon dioxide. Okay. Whereas C3 plants, okay, they are less efficient in carrying out photosynthesis than the C4 plants. Okay. So 2:1 is the answer for this question. The next question is in hatch and slag pathway, means they are talking about the C4 plants or the C4 pathway. So in hatch and slag pathway, the primary carbon dioxide receptor is, okay, options are oxaloacetic acid, phosphoglyceric acid, phosphoenol pyruvate, and last option is Rubisco. So Rubisco is an enzyme, so this is not the answer. And then we have oxaloacetic acid. Okay, this is the product. Okay, not the acceptor. Product of the what? C4 pathway or the C4 cycle. That's why it is known as the C4 cycle, because it is a 4-carbon compound. Phosphoglyceric acid, no, it is the product of C3 cycle. Okay, so this is also not the answer. Phosphoenol pyruvate. Okay, so this is the correct answer here. So what happens in C4 cycle? Carbon dioxide combines with phosphoenol pyruvate to form oxaloacetate, which is a 4-carbon compound. Phosphoenol pyruvate is a 3-carbon compound. Alright. So what should be the answer here? Phosphoenol pyruvate is the correct answer. Okay. So this was the end of this lecture. Thank you very much for listening.